Hello my dear students, welcome back to Biology at Fyojit. In today's class, we will discuss gastrulation in chick embryo, one of the most important topics in developmental biology and embryology. Gastrulation in chick is much more complex and prolonged than in frog. The process actually begins before the egg is laid and continues up to the second day of incubation. The most characteristic feature of chick gastrulation is the formation of the primitive streak, which acts as the main organizing center for the development of the embryo. Before understanding gastrulation, we need to know how the chick embryo develops during the early stages of its development. Early embryonic development in chick fertilization. In chick, fertilization occurs in the upper region of the oviduct. Only one sperm successfully fuses with the egg nucleus. After fertilization, the zygote travels through the oviduct for approximately 22 hours before the egg is laid. Interestingly, embryonic development starts while the egg is still inside the mother's oviduct. Cleavage in chick. Unlike frog eggs, chick eggs contain a huge amount of yolk. Because of this large yolk mass, cleavage occurs only in the small disc of cytoplasm present at the animal pole, known as the blastodisc. The yolk itself does not divide, therefore chick cleavage is called meroblastic cleavage, which is shown by the image, that is partial cleavage, and discoidal cleavage, that is restricted to the blastodisc region. This is the surface view of germinal disc of hen's egg showing early cleavage. This is the two cell stage, which is followed by 6 cell stage, then 20 cell stage, late cleavage stage, and this is the relationship between yolk and surface blastomeres. Structure of the blastodisc. The blastodisc consists of two regions: central region, which is whitish and circular and actively undergoes cleavage. Second region is the peripheral region or periblast. Central region is the actively undergoes cleavage. It is whitish and circular because it has no yolk material. But the peripheral region, which is darker in appearance and merges gradually with the yolk, it helps in nutrient transfer later. Cleavage stage. First cleavage. About 5 hours after fertilization, the first cleavage furrow appears. It is meridional but does not completely divide the blastodisc. Therefore, distinct cells are not yet visible. Second cleavage. The second cleavage occurs at right angles to the first one. Even now, clear blastomeres are not formed. Third cleavage. The third cleavage is vertical and parallel to the first cleavage. This stage produces 8 blastomeres, although the cell boundaries are still indistinct. Fourth cleavage. The fourth cleavage produces 8 central blastomeres, 8 peripheral blastomeres. For the first time, definite cells become visible. The central cells separate completely from the yolk. Formation of blastoderm. Further irregular cleavages produce a multicellular blastoderm. The peripheral blastomeres merge with the syncytial periblast. The periblast then plays an important role in transferring nutrients from the yolk to the developing embryo. Formation of subgerminal cavity. At cleavage continues, horizontal divisions occur. The central cells become 2 to 3 layers thick and gradually separate from the yolk. This separation creates a cavity beneath the blastoderm known as the subgerminal cavity, which functions as the blastocele. Formation of blastula or discoblastula. Continuous division finally produces a disc-shaped blastula called the discoblastula. It floats on the surface of the yolk mass. This is the image showing the step-by-step process of the formation of blastoderm, hypoblast, epiblast, etc. Area pellucida and area opaca. The blastoderm now differentiate into two regions. Area pellucida, the central transparent region, is called area pellucida. It is lifted away from the yolk and eventually from the embryo proper. Area opaca, the peripheral opaque region, remains attached to the yolk, that is called area opaca. Regions of area opaca. The area opaca is further divided into germ wall, margin of overgrowth, zone of function, etc. Formation of epiblast and hypoblast. We have studied in earlier image also. The blastoderm contains two types of cells: large yolk-rich cells and small yolk-pore cells. The yolk-rich cells move downward while the smaller cells remain on the surface. So as a result, upper layer cells form the epiblast and lower layer cells form the hypoblast. So the cavity develops between them is known as the blastocele, and the separation of these two layers, epiblast and hypoblast, is known as delamination. Gastrulation in chick. Gastrulation begins approximately 4 to 5 hours after incubation and is completed by about 22 hours. The process occurs in two major phases: formation of endoderm and formation of primitive streak and mesoderm. Formation of endoderm. The hypoblast contributes to the formation of endodermal layer. Several theories have been proposed to explain endoderm formation. Infiltration theory proposed by Peter in 1923. According to this theory, yolk-rich cells fall into the blastocele and migrate forward to form the endoderm. Delamination theory. This theory states that blastoderm splits into two layers. The lower layer forms endoderm, while the upper layer forms ectoderm. Involution theory. A slit-like opening appears posteriorly and cells roll inward into the blastocele to form endoderm. Invagination theory or Jacobson 1938, the posterior blastoderm folds inward and the invaginated cells become endoderm. Formation of primitive streak. The primitive streak is the hallmark of chick gastrulation. It appears in the posterior part of the area pellucida around 8 hours after incubation. The primitive streak develops due to the convergence of blastoderm cells towards the midline. As development proceeds, it elongates toward the anterior region. Primitive group and primitive folds. By the 18 to 19 hours of incubation, the primitive streak becomes well developed. A narrow depression appears in its center, known as the primitive groove. The elevated margins on both sides are called the primitive folds. Hensen's node. At the anterior end of the primitive streak lies a specialized structure called the Hensen's node. It consists of densely packed cells and contains a central depression called the primitive pit. Hensen's node function as the organizer of the chick embryo and is comparable to the dorsal lip of the blastopore in amphibians. Immigration of cells. Cells now move inward through the primitive streak. This inward migration is called immigration. These migrating cells form precaudal plate, notochord and mesoderm. Formation of mesoderm. The mesoderm differentiate into three major regions like dorsal mesoderm, intermediate mesoderm and lateral mesoderm. Dorsal mesoderm is located beside the notochord. It becomes segmented into somites. The first pair of somites appear around 21 hours and 4 pairs are present by 24 hours. Intermediate mesoderm located between dorsal and lateral mesoderm. By latter forms the kidneys and parts of the reproductive system. Latter mesoderm extends towards the periphery. It splits into somatic mesoderm and splanchic mesoderm. The cavity found between them becomes the coelom. Somatopleure and splanchopleure, two important embryonic combinations are formed. Somatopleure is the ectoderm plus somatic mesoderm, and splanchopleure endoderm plus splanchic mesoderm. Formation of notochord. Cells migrating from the Hensen's node form a rod-like structure called the notochordal process. This latter develops into the notochord. Meanwhile, the primitive streak gradually regresses and eventually becomes incorporated into the tail bud. Chick embryo at 24 hours. This is the image showing gastrulation is completed. Embryo becomes oval shaped. Primitive streak is fully developed. Notochord extends anteriorly. Head folds appears. Foregut develops. Mesoderm differentiate. 4 pairs of somites are present. Coelom formation begins. Blood islands appear in area opaca. Neural groove and neural folds become visible. Chick embryo at 36 hours. It foregut elongates, neural tube forms, brain differentiates into prosencephalon, mesencephalon and rhomencephalon. Neural crest cells appear, infundibulum develops, 13 pairs of somites are present, heart becomes tubular, blood vessels develop, primitive streak disappears completely. This is the image showing the chick embryo at 36 hours. Chick embryo at 48 hours, cranial flexure develops, embryonic torsions appears, 11 neuromeres become visible, brain constrictions are evident, extraembryonic circulation become complete, intraembryonic blood vessels form, 2 pairs of aortic arches develop, heart twist and chamber formation begins, blood circulation starts, Optic cup and lens vesicle appear. Auditory vesicle develops. 28 somites are present. Extraembryonic membranes grow rapidly and pronephros appears. So my dear students, to summarize the concept, gastrulation in chick is characterized by the formation of primitive streak, migration of cells through Hensen's nodes, and the establishment of three primary germ layers: ectoderm, endoderm and mesoderm. This process lays down the basic body plan of the chick embryo and prepare it for nudulation and organogenesis. Understanding chick gastrulation is extremely important for undergraduate and postgraduate examination, CSIR NET, SET, GATE and other competitive examinations in life science. See you in the next class. Thank you.